Now we will understand how nanoparticles can be used for delivery systems of siRNA. So you all know about siRNA, right? siRNA is basically very important when you are using them for regulation purposes. They are referred to as silencing RNA molecules. RNA interference is one of the fundamental mechanisms that we always discuss when we are speaking about gene regulation in any organisms. We very well know that whenever we want to control the expression of a specific gene, you have to interfere with the process of its expression. Wherein we know that if we convert an mRNA to a double-stranded RNA, it can obviously stop the production of a specific protein. This process of converting mRNA to a double-stranded format in order to stop its expression is what we call as silencing. In this way, you can turn off the production of specific proteins, and this has a lot of scope in terms of cancer therapy and also dealing with other neurological and metabolic disorders. This RNA interference mechanism is highly specific, and that's the reason why the discovery won the scientists the Nobel Prize of Medicine in 2006. RNA interference plays a very important role in switching the genes off during an organism's development or controlling the cellular functions as well. But the discovery of RNA interference not only enables the scientists to better understand the fundamentals of gene regulation, but it has also opened up a big arena of possibilities for genetic engineering. Let me tell you that silencing RNAs have been utilized to a great extent in therapeutics as well. But its delivery is something that is very crucial. It has to be delivered at the right site, in the right concentration. The biggest challenge with using siRNAs is that it has a very low stability in biological fluids. And somehow it has also been found to have low specificity of action due to off-target effects. To avoid all this, people have tried delivering the siRNA using nanoparticles. This was first done by researchers at iNano Aarhus University, wherein they developed nanocarriers for targeted delivery of silencing RNA molecules. They developed a novel chitosan-based siRNA nanoparticle delivery system for RNA interference both in vitro and in vivo. Chitosan is a naturally occurring cationic polysaccharide, and this has been used very widely in drug delivery systems in the past as well. It contains positively charged amine groups that can interact with negatively charged backbone of siRNA. So it's a perfect match to form a complex of nanoparticles and siRNA. These protonated amine groups, they allow the transport across cellular membranes and subsequently also enable endocytosis into the cells such that it is delivered into the cells very effectively. And it has been proved that chitosan siRNA nanoparticle delivery system has been very effective in silencing the genes both in vivo as well as in vitro. Very important to remember is that this particular technology is biocompatible, it's non-toxic and biodegradable as well. Please understand that there are some modifications also which have been done to this particular technique to make it more viable and effective. So you will see that, people earlier used to make use of polyl lysine carriers for delivering siRNA into the cells. But obviously that has been very toxic. It has had a lot of side effects, because of which people prefer nanoparticles like chitosan nanoparticles. But there are modifications which have been done so that these nanoparticles are retained into the system for a longer period of time. And at the same time, toxicity is at its least possible levels. The next thing that we are going to discuss is thermoresponsive systems. This is wherein you're trying to accumulate nanocarriers at specific site, like tumors, such that a lot of nanoparticles accumulate in that region and then you activate them using light, which generates a lot of heat. So when you activate nanoparticles, they respond by generating heat, right? And that brings about the effect of killing of tumorigenic cells, which we have discussed earlier as well. Now, let me tell you that these thermoresponsive systems are also very, very important, so that you aggregate all the nanoparticles at one site so that the response generated in terms of heat is higher. And also, once activated, their re-entry into the blood can also be prevented, okay, because they aggregate. So once they aggregate, their size becomes huge, and if their size becomes huge, they cannot now cross the cell membrane barriers and come out of the cell. So that aggregation is going to prevent its re-entry into the blood, which is very, very significant, obviously. You can make use of nanoparticles in all different formats. You can make use of it as mishls, nanoemulsions, nanospheres, nanotubes, fibers, liposomes, dendrimers, and even hydrogels. In the next part, we will see how we can make use of nanoparticles for theranostics, which is a very, very important area, and also to understand how we can do tissue engineering using nanoparticles.